Hi, welcome to biology classes with Lakshmi Chandra. We are dealing with a series of sessions on translation in prokaryotes. After completing the overview and pre-initiation or charging of tRNA, in today's session we'll focus on the next most important stage of translation in prokaryotes, that is initiation. How actually this initiation is happening. So as told in the previous sessions, here I have just compared the different key players to cinema. That is, mRNA is the heroine here, who is there sitting in a factory named as ribosome. Okay, and she's guiding all the activities there for the synthesis of proteins. She has got so many codons with her, and with the help of the information present in those codons, she is directing the hero, that is tRNA here, to bring the respective amino acids. And the other members, that is the enzymes, the initiation factor, the elongation and the release factors, they together are the co-workers over here, the supporting staff, and they are actually doing the whole process of protein synthesis. So in today's session, we will be dealing with what is the initiation process, how this factory begins to do the process. For that, we should first understand regarding the factory, that is the ribosome. Regarding the ribosome, a clear understanding was given in the overview session, in that session, whose link is given in the description box. You may go through that. Anyway, let's have in a nutshell, let's look into what is this ribosome actually. So ribosome, in the case of prokaryotes, it is a 70S ribosome which is having two subunits. A very small subunit it is having. This subunit is called as the 30S subunit of prokaryote, and this 30S subunit is having one small 16S rRNA here. So let me just write that 16S rRNA. I am representing like this. This is 16S rRNA, okay. This is part of 30S ribosome. You know, 30S ribosome is made up of many rRNAs. So among that, one rRNA is 16S rRNA. It is having a sequence here, UCC, UCCA. This is the sequence present along with 30S subunit. 16S rRNA part of 30S subunit, it is present. And to this sequence actually the mRNA, that is the heroine, okay, having the codons, that is the information to produce the proteins, it comes and sits, binds actually here. This is, I am just drawing the mRNA with this red line. So here, naturally, when they have to bind, they have to have complementary sequences. So here the complementary sequence is A with U, G with C, again G with C, A with U, G, G and U. So this sequence will be there with mRNA, and it is this sequence which is actually aiding the mRNA to come and bind with the small subunit of the ribosome. That is very essential for the process to begin, for initiation to happen. And this sequence is given by a name. It is called as the Shine-Dalagarno sequence. So for initiation, you all should understand that it is the Shine-Dalagarno sequence which is actually helping this mRNA to come and attach to the exact part of the small subunit of ribosomes. Okay. Thereafter, there can be many other different units present, different bases present, and then comes the AUG codon. AUG codon. I hope you remember the importance of AUG. It has got a dual role. What is the first role of AUG? AUG codon codes for the amino acid methionine. And AUG is also the start codon. That means initiation of translation begins with the advent or with the coming of this codon, AUG. AUG is the start codon. It also codes for the amino acid methionine. This is a dual role. So regarding this codons, the different codons present, how they are arranged, and what amino acid each one of them codes for was actually dealt with in the previous session. Its description is also, its link is also given in the description for your easy understanding. You can just click onto that and watch that for having a clear understanding about it. So here AUG is having these two roles, and since we are dealing with the prokaryotes, always remember the initiation codon here is coding for not for methionine, but it is coding for formyl methionine. Formyl methionine, or in short it is called as FMET, formyl methionine, FMET. So usually AUG is coding for methionine, whether it is starting or in between, wherever is this AUG coming, it is coding for methionine only. Okay, but for prokaryotes alone, the initiator AUG is not coding for methionine, but it is undergoing a bit of modification, formylation and all. It is added with formyl group, and it is formyl methionine is the group which is coming as the initiator amino acid. And so the tRNA which is carrying it for the first time will carry the formyl methionine. That amino acid it will carry. It will not carry the normal methionine in the beginning, in the initiation. Okay, and as I have told before, this small subunit is having three areas: E site, P site and A site. Okay, so A site is the region where it is the acceptor site or amino acylation site, or this is the area where actually the tRNA which is carrying the amino acid comes and sits. This is the entry site, acceptor site. But as I have told you, the first tRNA is coming not with methionine, but it is coming with formyl methionine, the first tRNA. So the first tRNA will not sit here in the acceptor region. Instead, the first tRNA which is coming will sit in the P site, peptidyl site, only the first one. The rest, whatever is coming, will be first sitting over this one and then moving. That movement I'll tell you. Okay, so imagine, so understand that the first tRNA that is coming is actually formyl methylated tRNA, and that will occupy the P site of 30th subunit of the ribosome. Now let's understand it in a deep manner. Let me just rub this diagram and I'll prepare a new diagram for you. Yes. So 30th subunit, let me just draw once again. This is 30th subunit. It is having E site, a P site and an A site. Okay, so as I have told you, tRNA, the first tRNA should come and sit over the P site. And for it to come and sit over the P site, the A site has to be blocked. Otherwise, it will come and sit in the A site only. Okay, so this A site is blocked by the initiator factor 1, IF1. IF1, the initiator factor IF1 will bind over this A site, and so the tRNA coming with formyl methionine cannot occupy this site. Okay, now this E site is again occupied by another initiator factor, that is IF3. IF3 will occupy the E site. Okay, so this IF3, when it is occupying the E site, the large subunit of ribosome cannot come and sit over the small subunit of ribosome. and the area is open. It is fully free for this tRNA to come and sit over the P site. See how wonderfully it is mechanized. Okay, so usually in our body, ribosome occurs like this as a combination of large subunit and a small subunit. But at the time of protein synthesis, they just divide themselves into two parts, but again they have the tendency to come and bind again. Okay, but then if they come and bind again, the tRNA cannot come and sit over here. It is very difficult. So what happens? For this initiator tRNA to come, and this initiator tRNA is carrying formylated methionine, F-met, that is the amino acid present. This I have drawn as tRNA, okay? And for this tRNA to come and sit over here, it has to have a methionine group exactly at the P site. So E and A site should be occupied by some other thing. Otherwise it will go and occupy that area. Fine. So what is happening? Who is helping that? IF1 and IF3. So IF1 will occupy the A site, IF3 will occupy the E site. IF3 is actually preventing the large subunit of the ribosome to come and bind over here. Now this P site is free, F-met will come and sit over here. So this I told you methionine. So if it is methionine, naturally this is mRNA, okay? 5' to 3' direction always it happens. So mRNA, the codon is AUG. The codon is AUG and this is the initiator codon. So the amino acid coming is methionine. Since it is initiator, it is formyl methionine. My question is, what will be the anticodon? Anticodon is there in the tRNA. So anticodon is complementary to the codon. So it will be UAC. It is UAC. So what is happening is, in the cytoplasm there is a tRNA having UAC as the anticodon. This tRNA understands that it has to come and sit on the P site and it has to come with the amino acid F-met. It has to come with the amino acid F-met and sit on the P site. But you know, this tRNA cannot on its own come and find the place and sit. It needs energy. It needs energy for itself to come and sit on this P site. So who is giving that energy? It is the GTP which is giving the energy. The GTP is actually giving the energy for F-met. But GTP on its own cannot combine with this tRNA and give it energy. For that, another initiation factor is required, that is IF2. So this IF2 combines with GTP and this GTP IF2 combines with our tRNA carrying F-met amino acid, and now they come and sit over the P site exactly. tRNA can now go and sit in the P site. On its own it cannot go. It needs energy. And the energy molecule needs the assistance of IF2. So here actually it is not alone, but it is combined with GTP, and GTP is combined with IF2. This is how the tRNA, the initiator tRNA, comes and binds. Once the initiator tRNA comes and binds, this GTP will be hydrolyzed or it will be converted to GDP. Once the GTP is converted to GDP, these factors, that is IF3, okay, IF1 and IF2 leaves this space. It leaves the space. Its role is over. It leaves the space. So when does that happen? Once GTP is converted to GDP, once energy is getting converted, it is passed on to this particular whole thing. All these initiator factors, IF1, IF2 and IF3 initiation factors, all of them, they dissociate from the ribosome, from the tRNA. They dissociate from here. So see here, now IF1 is not there. So who can come? Yes. Now the large subunit is very happy. It can occupy the space. The large subunit has come. It can occupy the space. Okay. So now A site is free. There is no initiator factor. So another tRNA can come and attach over here. So now since the large subunit has come over here, we can say that the whole thing, that is the initiator complex, is now ready. Initiator complex. The initiator complex is now ready. So the process of initiation ends here. Next, as I've told you, the A site is free to receive another tRNA with an amino acid, and after it receives, what is happening inside this factory of ribosome, how the process is actually, the protein is actually synthesized, that we will definitely see in the next session. So if you want to have clarity, please click on to the links given in the description box to refer to the previous videos. Thank you all.